I'm going to hand it over to Marthe, and I are going to tag team this presentation, and Marthe is going to start off, and then I'll finish up. She's going to go into a lot of the underlying statistical methods, and then I'll share some actual business use cases that reinforces, you know, the critical importance really of applying these statistical methods. But Marthe, why don't you go ahead and start?
Yeah, absolutely. Thank you. Thank you for the introduction. So yes, what we are going to show today is actually the statistical methodologies to detect data anomalies in clinical trials, and we'll do that mainly through some illustrations. So I'll start off with a made-up, very simplified illustration, but later on in the presentation, as Steve just said, there will be some real use cases as well. So I want to start off with this example just because it makes a few points. So in this case, what you can see is that we've actually depicted 9 centers. So every square is a center. So we have center C1, C2, up to C9. And we see here the data from the systolic blood pressure over time. So the vertical axis is the systolic blood pressure, and that's then collected over time. And you can right away see a few centers that show some data anomalies as well. So for example, C3 clearly has some different data patterns than the other centers, which is a lack of variability. So if we look at here, each line is one patient, right, within the center. And you can clearly see that every patient has a similar systolic blood pressure. So it starts from a certain value, it goes down, and then it goes up again for every patient. Center C7 has a completely different kind of issue, but still also striking, is still a line per patient in the center, but now the patients don't have the exact same values as each other, but they do have the exact same values across all the visits, so they're propagated, as we call it. And then finally, this is an interesting one, this is a shift in mean, so you can clearly see compared to all the other centers that this center C5 has a higher mean overall than all the other centers. Now, when we say data anomalies, that doesn't mean that there's necessarily an issue. Like this might be potentially a specialized center interested in hypertension, for example, then this might not be an issue, but it could be. It's worth investigating. There is a data anomaly. All the examples I showed were univariate, right, looking at one variable at a time, but of course the power also lies in looking at multiple variables at the same time. An example could be a correlation. So if we stick to that systolic blood pressure example, but we also add diastolic blood pressure to this, let's say that this is the expected correlation across the patients here, and then we look at a certain center X, as you can see there, the correlation is almost perfect. Again, this doesn't necessarily mean that there is an issue, but it is suspicious, so it's worth to further investigate. Now, all those ideas aren't new. So actually 20 years ago, the founder of CluePoints, Mark Buys, guided actually a group of statisticians to create a paper that listed everything until then to detect potential fraud in clinical trials. So that paper was kind of a summary of the state of the art at that time. So that listed all those tests for one variable at a time, but also several variables at a time. And what it also did was, for example, how we can use repeated measurements, because of course that is a rich amount of data to investigate. And another topic was the calendar time. So of course we can do also a lot of things with dates and times, as Steve will illustrate later on. So what is new then? At CluePoints, what we try to do is actually come up with a software that implements all those different individual ideas into one software. And what the software is aiming to do, and in general what central statistical monitoring is aiming to do, is looking at your clinical trial data from a statistical perspective. And of course, you know the importance of central statistical monitoring based on the FDA guidance and the EMA guidance. But basically, what you want to make sure that your central statistical monitoring is doing, and what we do with CluePoints, is that our methods are unsupervised. So unsupervised for us is no prior assumptions are needed, because actually you should assume that you don't know what you're looking for. You don't know where your issues are necessarily. Now, in some cases you might, and then you want to do some specific analysis, of course, but overall you don't necessarily know where your issues are. We also want to make sure that our methods are agnostic. Agnostic in a sense that we want to use generic statistical tests not requiring any context knowledge, meaning that as much as possible, we can apply our methodology to clinical trials from cardiology or oncology or any other kind of therapeutical area. The most important thing I would say for the CluePoints methodology, at least, is that it's exhaustive. So we really look at all the data regardless of importance, and we submit it to a huge amount of tests. And a lot of the methods out there are really focused on some primary endpoints, some very important variables in the trial, like primary endpoints, secondary endpoints, sometimes maybe some safety endpoints. But at CluePoints, we really try to look at all your data. Of course, looking at some of those endpoints is very important, and you can do that as well. But it's every now and then it's very good to also look at exhaustively at all your data in the trial, because you will see in the examples from Steve later on, actually, that some of the issues, the anomalies that were flagged actually come from less important variables in the trial. We also want to make sure that our tests are robust. So our tests require minimal distributional assumptions so that we shape our data in such a way that we don't have to deal too much with those distributional assumptions. And then finally, we want to make sure that our tests are sensitive and specific, of course. So we want to make sure that what we flag is indeed an issue, but at the same time that if there is an issue, that we're able to flag it. To be completely honest here, in the beginning, we were flagging too many things. So we had too many issues flagging, and that was actually because we weren't allowing for natural variability at the moment. So for an example there is we were comparing centers with each other on weight, and there were a lot of centers flagging, and that was because we didn't take into account some natural variability that happens just because centers are located in different countries. So we're using mixed effects models, for example, to allow for that natural variability. So what comes out of CSM and what comes out of CluePoints is actually a large matrix of p-values, all those p-values from all those individual tests. So at CluePoints, how we do it is based on the type of the variable, so whether your variable is a continuous variable or a categorical variable, but also based on the type of whether your variable is collected only once or repeated over multiple measurements, we're going to apply a numerous amount of tests, let's say 5 to 10 statistical tests. If you then know that you have in a typical average clinical trial, 20 to 100 clinical sites, and we have 250 to 1,000 variables to test, that adds up to a huge amount of p-values. And how we organize it is in a matrix of p-values, and this is here an example. So how this is organized is one row per center, and then one column per test that is done. So for example, center 2 had a mean test done on the labs data set on a variable lab 1. And what they say is this is actually all the p-values that come out of it. You will see, of course, that you have some negative values. A probability cannot be negative, but this shows just the direction of the effect. So in this case, if it's negative, it means that the mean in center 1 was lower than what we expected. If it's positive, it means that the mean is higher than what we expected. Now, all those p-values are good, are interesting, but in a practical way, what do you do with them? So where the power lies in, at least for CluePoints, is what we call our data inconsistency score. So what we're going to do is we're going to basically average all those p-values in a summary score, which is, let's say, our p, and then we're going to take the minus log 10 of that average, which comes to what we call our data inconsistency score. And actually, when you use that kind of transformation, you get very close to a similar kind of scale that is used for the magnitude of earthquakes, so the Richter scale. So for the magnitude of earthquakes, of course, you all know that that goes from 0 to 10. 0, there's nothing. 10, basically, the country is destroyed. And similarly, with the data inconsistency score, we also get to a kind of scale that goes from 0 to slightly above 6. So we say everything that has a score of lower than 1.3, that's not statistically significant. So that's not necessarily something to look at that much. Everything that is between 1.3 and 2, that becomes minor significance. So that's a 10 to the minus 2 p-value, and so on. Now, what do we do with that data inconsistency score? Of course, we are going to use that to identify our outlying centers, as we call it. And we visualize it using the bubble plot. So this is a bubble plot. And what you see here is that every bubble here is a center. And the size of the bubble also reflects the size of the center. So in this case, this is a big center of like 42, 43 patients here. The vertical axis lists a p-value or this inconsistency score. So the higher you are ranked on here, the lower your p-value, so the higher your score. So the more atypical your data is compared to all the other data. So we flag here 3 centers as being different from all the other centers. So our tool is really meant to rank centers in how different the data is compared to all the other centers. What you can see is that this center 18 is ranked 1, so it's the first atypical center globally, looking at all our data with a size of 15 patients. What does this mean is that the probability that this center's data, taking into account all the data, of course, is consistent, compatible with the data from all the other centers is only 10 to the minus 6, right? So very low probability. At this moment, we don't necessarily know why. We don't necessarily know what the issues are or whether there are even issues. We just flag that this center is different from all the other centers. And then we have to go and start to explore to figure out what the issues are exactly. And that's what Steve is going to tell you more about, like the kind of issues that you can find whenever you have detected indeed those atypical centers. So Steve, with that,
excellent.
I'll leave it up.
No, thank you very much, Marthe. So, yeah, as Marthe said, you know, so you can see from how we compute the overall inconsistency score, it provides a very easy entry point for study teams to very quickly identify sites that they need to pay attention to and investigate further. And you know, it would take more time than we have today to show you, but it's a very user-friendly interface that takes you down to the details and allows you to then quickly understand what is going on at those sites that are atypical and what the potential issues are. You know, as Marthe said, you know, the founder of CluePoints back in the 1990s was originally looking into and primarily focused on the science of fraud detection in clinical research. What we have found and our customers have found over the years is that these methods are effective not just at detecting fraud, but all types of potential data quality and study misconduct issues that don't necessarily mean fraud. So the types of issues, I have 3 categories listed here: systemic type of errors, completely unintentional. They could be just simple site training issues that are leading to unreliable data patterns, or it could be miscalibrated or misconfigured broken study equipment taking measurements. It could be sort of tampering or sloppiness. We see a lot of propagation or lack of variability in various vital signs, underreporting of adverse events or con meds, some procedures or assessments not being performed when they should at a higher rate than other sites. And then, of course, we still do find fraud, unfortunately. And as I've told people, since I've been at CluePoints the last 4 years, there's more fraud out there than I would have expected before that in my career. It's unfortunately, it happens. It is more rare though than all of these other types of issues. Next slide, Marthe. So let's take a look at a few examples just to illustrate the point. And these are real examples of issues that our clients have found on various studies. This is an example of a systemic error. It was actually miscalibrated thermometers. It was a large global study, a vaccination study, so patient temperatures were being taken repeatedly for each patient. It was a relatively important measurement on the study. And in addition to running the statistical test at to compare sites, you can also compare countries, and that's what was done on this trial as one of the approaches. And you can see that all the patient temperatures in Spain were in a slightly lower range, and it was the mean test. So it was one of the suite of statistical tests that caught this and led to an investigation which discovered that all the thermometers shipped to Spain for that study were miscalibrated the same way, leading to this offset. So an important finding, but not fraud, just an unintentional miscalibration of equipment. This one is more in the middle. It's that tampering sloppiness type of issue, and in this case, it's patient weights. Each graph there are patient weights measured over time for different centers, and you can see we've focused on center 6 where you can visually see that there is a strange lack of variability between patients over time. And this was identified with a couple of our statistical tests, looking at statistical variability in measurements across patients and within patients. And what the site ultimately admitted is that they were interpolating weights between visits and not necessarily taking the weight measurements at every visit for patients. So, you know, it was a tampering. And of course, this is one now of actual fraud. This is a view from the CluePoints application for a specific site. It's sort of a drill down from that bubble plot that Marthe showed you a couple of minutes ago. Each lane, all the dots are individual test results, and each row in the view is a different domain of data. And we've circled the dots further to the right represent more atypical. So the more atypical test results. And you can see all the way on the right, we've circled one of them. It was a key finding on this study, and it had to do with ePRO diary data. The patients were all entering daily entries, and for one site with 15 patients, one of the tests was on time of day for the diary entries, and it was very, very statistically unusual. We were seeing over 70% of the daily entries in the same hour of the day. In fact, at the end of the business day, in the 6 p.m. hour. And if you click Marthe, what we found is that when the study team followed up, they finally got the site to admit that they never handed the diary devices out to those patients. At the end of every working day, one of the study nurses would pull the devices out of a drawer and invent diary entries for those patients. Not something that the site monitor found, not something that the data management team or other reviewers found. And it's a theme throughout all of these examples that's really important that I want to reinforce, which is our traditional methods of quality oversight and monitoring very likely would not have found these issues that we're showing in these examples. And that's what we're seeing all the time on the studies that are in CluePoints, is that, you know, the statistical monitoring gives a perspective that even really, really good site monitors just don't have the context to be able to see those patterns and the differences. So it really reinforces the critical importance in overall data quality management of these statistical methods and central monitoring in general. You go to the next slide. I'm just going to go through this one really quickly. This is a very recent example. We did what we called a CluePoints Data Quality Challenge, where we invited organizations to bring us studies that were previously conducted that they have known significant issues in, quality issues, and they fed us the study data. We ran it through CluePoints to see if we could identify where those issues were. And we were blinded. They didn't tell us anything about what the actual issues were or where they were. And in fact, when we ran CluePoints on this recent exercise, we identified 6 very atypical sites. One of them, one of the highest enrollers, site 105, they revealed to us after we ran the analysis that that was the site that had the major known issues that they discovered late in the course of the trial. The CluePoints analysis findings matched very closely with what the study team actual observations findings were for that site in the study. So it really reinforced the power of these statistical methods to identify and expose problems. On that particular study, this site was discovered as a big problem pretty late in the course of the study. And what we did on this exercise is we created snapshots of the study database at earlier time points, and we went progressively earlier. And what we found was the same site, site 105, was the most atypical site in the analysis, all the way back to a 5% progress point of that site's participation in the study. So if this study team had used the CluePoints central monitoring and statistical monitoring methods, they would have clearly identified this site many, many months earlier and may have avoided a lot of impact on the study. That table just shows an estimate of the direct cost of actually managing Site 105 for that additional time that was lost. So there's direct cost implications, but the bigger implications are potentially needing to do extra recruitment to replace the 90 or 100 patients at that site that they couldn't use in the analysis because they were unreliable. Or the increased risk of not having enough power to prove key study endpoints because of the removal of those patients from the study. Both of those potentially much bigger implications from a business perspective, delayed and or no marketing approval for the drug. So very important. Just in general, looking at, these are just a few examples of what our customers find all the time. It really reinforces, as I said, the importance, the criticality of having an effective centralized monitoring solution in place. It doesn't mean you're replacing other traditional methods of review. Clinical data management still plays an important role in data quality. Medical safety reviewers play an important role. Site monitoring still plays an important role. We could have a whole separate long discussion about how much SDV and SDR should be done, and of course it should be more targeted in our view and much more limited. But there's still importance in that site monitoring function. But centralized monitoring is a critical component of quality oversight, and often finds things that are more impactful and broader and more important in scope than our traditional methods find, because they're more transactional in nature, manual reviews of things, record by record inspection, rather than this holistic statistical approach, which we've proven finds much more impactful issues out on your trials that those traditional methods are often not able to detect or identify. At CluePoints, in addition to the statistical methods, we have been actively investing in and adding techniques based on machine learning and other predictive analytics techniques, artificial intelligence. We already have in our platform, on top of the statistical methods, machine learning techniques that are effectively automatically grouping sets of test results that represent thematic issues based on past issues that have been discovered. So it's using the learning from the hundreds of trials of data that we have in our database to actually present more efficiently and quickly to central monitors. Here's the group of results that you should be looking at together as a potential issue. We're also very heavily investing, not just for the statistical tests, but for KRIs and QTLs, into predictive analytics, methods that are going to allow teams to see an emerging risk much sooner and be able to see that maybe something that was an issue, the site is now improving on, and be able to detect that improvement has or has not occurred efficiently and quickly. So in summary, central statistical monitoring is a very powerful tool for quality oversight. Early detection is a key to success, and as I was just mentioning, we've been investing for years, not just in effective and reliable detection of risk and issue, but optimizing for earliest possible detection of issues as well. And of course, as I mentioned with that use case, early detection is a key. It's not just about detecting risk. It's about detecting it as early as possible so that you can prevent significant negative implications in terms of patient safety, the integrity of trial outcomes, potential delays that may be, and as I mentioned in that example, direct study costs. So we got 5 minutes left. I'll stop there. That's the end of our presentation. And if we have any questions, Eric,
Yep.
Marthe and I are there.